So today we will start the metabolism of lipid. Okay, and as you know, the main lipids are fats, includes fats, cholesterol, and phospholipid. And in fact, in this chapter, we mainly talk about the metabolism of fats and cholesterol. Okay. So first, let's see. I will generally introduce you the structure and function of fats and cholesterol. The storage form of fat in mammalians is triglyceride. Okay, and this figure shows you the structure of this triglyceride, and the triglyceride is composed of glycerol, and this glycerol is esterified with 3 fatty acids. Okay, so this is the structure of the triglyceride, and the fatty acid of the triglyceride can be divided into the saturated fatty acid and unsaturated fatty acid, which depends on the number of the double bonds in the fatty acid chain. The saturated fatty acid means there is no double bonds in the fatty acid chain, and this saturated fatty acid mainly derived from animal sources. And another is the unsaturated fatty acids. And unsaturated fatty acids means that there are a number of double bonds in the chain, and which are mainly derived from some plant origin, such as sunflower, soybean, or corn oil. And these unsaturated fatty acids can be divided into mono unsaturated fatty acid and polyunsaturated fatty acid, which depends on the number of the double bond in the fatty acid chain. The mono unsaturated fatty acids have only one double bond, and these mono unsaturated fatty acids are found mainly in the olive oil and rapeseed oil. And the polyunsaturated fatty acid belong to the essential fatty acids, which cannot, okay, here you should remember an essential fatty acid. That means that this unsaturated fatty acid cannot be synthesized by the body and need to be taken in the diet. Okay, and the essential fatty acids includes linoleic acid and linolenic and arachidonic acids. This means arachidonic acids, okay, and the linoleic acid includes 18 carbon atoms and 3 double bonds, and this the linoleic acids contain 18 carbon atoms and 2 double bonds, and this linolenic acid includes 18 carbon atoms and 3 double bonds, and this arachidonic acid includes 20 carbon atoms and 4 double bonds. Okay, and the first two, the linoleic acid and linolenic acid, mainly as structural components of cell membranes, and the third one is arachidonic acid, is a precursor of the prostaglandins. Okay, so here you should remember that what is the essential fatty acids. Okay, that means that we cannot synthesize by the body and needed to be taken in the diet. And you should remember which fatty acids belong to the essential fatty acids. Okay, so another very important lipid in our body is cholesterol. Okay, so the cholesterol is a precursor of sterols. And this figure shows you the common structure of sterols. Okay, this is a cyclopentene. The common structure we named as a cyclopentene and polyhydrophenastyrene. Okay, and cholesterol is taken in the diet if meat or animal products are consumed. So it's not present in strict vegetarian diet. So it's not a dietary essential because the cholesterol can be synthesized in our body. Okay, so next we will let's see the digestion and absorption of fat. Fat can be digested by lipids, but as we know, fat is insoluble in water, right? So here comes the question: how fats be digested by the water-soluble lipids? Here, this figure shows you the digestion of the fat. Okay, and because the fat is physically unwieldy in an aqueous media, and as lipids, okay, here as lipids can, and as because lipids can only attack at the oil-water interface. Here is a this one is a this linear means the oil-water interface, okay? So the area of this interface in a simple fat-water mixture is insufficient for the required rate of digestion. So how to solve this problem? The available surface area is increased by the emulsifying the fat. That we call this process is emulsification. And the bile salts, here you should remember, the bile salts plays an important role in this process, emulsification. So the bile salts, the bile salts can act as a biological detergent to help to emulsify the lipid into the droplet. Here you can see, okay. And the bile salts are synthesized. Usually, the bile salts are synthesized in the liver and stored in the gallbladder until discharged into the duodenum. Okay, and then the bile salts can act as a biological detergents to help to emulsify the lipid into the small droplet. Okay, and after emulsification, the lipid is converted into the small droplet, and as a result, the available surface area is increased for the further digestion, right? Okay. And the main digestion of fat occurs in the small intestine by the action of the pancreatic enzyme lipids. And lipids can catalyze the triglyceride to the monoacylglycerol and fatty acid. And then the products of digestion of fat can be absorbed into the intestinal cells by resynthesized into fat. That means the fatty acids can be re-esterified to produce the triglyceride. Okay, and then the triglyceride together with the absorbed cholesterol must be transported from the intestinal cell to the tissues of the body. Triglyceride and the cholesterol cannot diffuse out of the cell through the membrane because they are insoluble in the blood. Okay, so the solution is that. So how to solve this problem? So the fat, that means the triglyceride and cholesterol, can together with other several proteins arranged in the epithelial cells into the particles. We call this particle as chylomicrons. And these chylomicrons can be released from the cell by the exocytosis, since they are too large to transverse the membrane any other ways. And usually, after a fat-containing meal, this lipid protein, the chylomicrons, enters to the bloodstream and gives the blood a milky appearance and increase the fats and cholesterol levels temporarily. Okay. So this is the digestion and absorption of the fat. Okay, so here I will show you a small reduce the protein, right? The digestion of the fat, for example, there involved a lot of enzyme in the digestion this nutrient, right? For example, in nutrient the fat, carbohydrate and protein. But in the digestion of the during the digestion of the fat, not only enzyme involved in this process, but only here is another very important cells is bile salts, right? The bile salts play a very important role in the digestion of fat. The bile salt can help to break down the big fat into the small droplet. Okay, so here you should remember. Epithelial cells can release the glucose and amino acid into the blood, right, without any change. But in the absorption of fat, this fatty acid or these fatty acids or the monoglyceride can be resynthesized in the epithelial cells to fat and then released to the blood as chylomicrons. Okay, so here is the difference. So as you see here, the digestion and absorption of fat is has some difference with the carbohydrate and protein. Okay. So I will check if everything goes well. Okay, next let's see the fat catabolism, that means lipolysis. Okay, the lipolysis starts from the fat mobilization. Okay, so first let's see what is the fat mobilization. The fat mobilization in fact describes a process that free fatty acids are released from the triglyceride through the hydrolysis by lipids, okay, in the adipose cells. And this slide shows you the process of the fat mobilization. And here you should first notice that the fat. You should remember that fat mobilization mainly occurs in the adipose cells, okay. And in the adipose cells, there is a very important enzyme is hormone-sensitive lipase, and these hormone-sensitive lipase can be activated by the glucagon and adrenaline. Okay, and this enzyme, hormone-sensitive lipase, is a key enzyme in the fat mobilization. So the triglyceride can be hydrolyzed by this hormone-sensitive lipase and release one molecule of free fatty acid and to produce the diacylglyceride. Okay. And then this diglyceride is further degraded into the glycerol and then results releasing two molecules free fatty acid by lipase. And the activity of this hormone-sensitive lipase is controlled by the phosphorylation. The phosphorylated enzyme being is the active form. Okay, and this is a fat mobilization. And here you should remember what is a fat mobilization. It's a process of free fatty acid and released from the triglyceride through hydrolysis by the hormone-sensitive by the lipase. Okay, and the fat mobilization is occurred in the adipose cells. Okay, you should remember. And then after fat mobilization, okay, after fat mobilization, the triglyceride can be hydrolyzed into glycerol and FFA means the free fatty acids. Okay, and next let's see how to digest it, how to catalyze the glycerol and the free fatty acid. First, let's see the glycerol. The glycerol can be further phosphorylated and oxidized to the dihydroxyacetone phosphate. And this dihydroxyacetone phosphate is an intermediate in the glycolysis, okay? And in the liver, in fasting and starvation, the glycerol can be converted into the glucose by the gluconeogenesis pathways. But this cannot happen in the adipose tissues. Okay. And then a fatty acid. And then next, let's see the metabolism of fatty acids. First is the activation of fatty acids, and this is the activation reactions, and the enzyme catalyzed this reaction we named as acyl coenzyme A synthetase. So, if we want to further oxidize the fatty acids must first be converted to an active intermediate form before they can be digested. Okay, and this is the only step requiring energy from the ATP. Okay, in the complete degradation of a fatty acid. And in the presence of ATP and coenzyme A, the enzyme acyl coenzyme A synthetase can catalyze the conversion of a fatty acid into an active fatty acid, or we name it as a fatty acyl. Occurs carried out in the cytoplasm. Okay, that means the activation of fatty acids. This reaction occurs in the cytoplasm, and then this activation of fatty acids should transform from the cytoplasm into the mitochondria. Okay. So next, let's see the transporter of fatty acyl coenzyme A. As we know, the mitochondrial membrane have the two, right? The outer and the inner, okay? The outer mitochondrial membrane and the inner mitochondrial membrane, right? And they have the different characters. The outer mitochondrial membrane is permeable to most metabolites, okay? As you see from this figure, the up one is means the outer mitochondrial membranes, okay? And this means the inner mitochondrial membrane. So the out mitochondrial membranes is permeable to most metabolites. So the fatty acyl coenzyme A can cross the out mitochondrial membrane into the intermembrane space. As you see here, this is a acyl coenzyme A synthetase, right? And as you see, these enzymes located in the outer mitochondrial membrane, right? And this enzyme can activate the fatty acid into the activation fatty acid. That means the acyl coenzyme A. And this acyl coenzyme A can cross the outer mitochondrial membrane into the intermembrane space, okay? And then because the inner mitochondrial membrane is selective to most metabolites, this is totally different from the outer mitochondrial membranes. The inner mitochondrial membranes is selective to most metabolites. So the fatty acyl coenzyme A cannot cross the inner mitochondrial membranes to reach the mitochondrial matrix. So how to solve this problem? Okay, and here there exists a special transport mechanisms on the mitochondrial membranes. Okay, and we call this the carnitine. We call these mechanisms is a carnitine transporter. Okay, so external face, okay, of the inner membrane. The acyl, as you see here, the acyl can be transformed from the acyl coenzyme A to a carnitine, okay, and this carnitine and then be catalyzed, and this and then the carnitine can be converted into the acyl carnitine, and this reaction is catalyzed by the carnitine palmitol transferase, okay, transferase 1, and this enzyme is located in the outer mitochondrial membrane, and then the fatty acyl, this acyl cartinase complex can be transformed across the inner membrane into the mitochondrial matrix here, and this translocation is carried out by this translocase, okay, and this translocase is located in the inner mitochondrial membrane, and after entering to the matrix, this carnitine is released, and coenzyme A is transformed to the fatty acid by the enzyme carnitine acyl transferase 2. And this transferase 2 is located in the inner mitochondrial membrane. And then the free carnitines can be returned to the intermembrane space by the translocase, and where it collects another fatty acyl group. Okay. So this is a process of the transport of fatty acyl coenzyme A from the cytoplasm to the matrix. And here you should remember that this, in fact, the transport of fatty acyls. Okay. So okay. And then after entering the matrix, the fatty acyl coenzyme A can be oxidized to the carbon. That means the conversion of fatty acyl coenzyme A into the acetyl coenzyme A molecules inside the mitochondria by this process, by a series of reactions, and we named this reaction as a beta oxidation. Okay, and in beta oxidation, 2 carbons are cleaved from the acyl coenzyme A molecules at one time. Okay, and the chain is broken between the alpha and beta carbon atoms, so we named as a beta oxidation. And next, we will see the reactions involved in the beta oxidation. Okay, so we start from this part. Okay. The first step is this is the acyl coenzyme A. Okay, the first reaction is to removal of 2 hydrogen atoms from the alpha and beta, or 2 and 3 carbon atoms. And this reaction catalyzed by this enzyme, acyl coenzyme dehydrogenase. Okay, and this reaction requires FAD as a coenzyme, and this results in the formation of the transaminal coenzyme A, and also form the FAD reduced coenzyme FADH2, and then the FADH2 can enter the phosphorylation chain, right? Okay. And undergoes further dehydrogenation, okay, on the 3 carbon of the beta carbon catalyzed by this reaction, the 3-hydroxyacyl coenzyme dehydrogenase, and to form the ketoacyl coenzyme A. And this reaction here, you should notice that this dehydrogenation requires the NAD as a coenzyme, and then the NAD can be reduced to the NADH. Okay, so after oxidation, hydration and oxidation, the beta carbon atoms is oxidized and is converted into a ketone group. And finally, in the fourth reaction, in the fourth step, this 3-ketone acyl coenzyme A is split at the alpha and beta position, or 2/3 position by the sialase, and then result forming acetyl coenzyme A and a new acyl coenzyme A. And as you see, compared to the original acyl coenzyme A, this new acyl coenzyme A with 2 carbon shorter, right? With 2 carbon shorter than the original acyl coenzyme A molecules. And then this new acyl coenzyme A formed in the cleavage reaction can be re-entered to the oxidative pathways. And when the fatty acid group has been shortened to the 4-carbon stage, okay, 4-carbon stage, that means the fatty acid chain includes only 4 carbon atoms. And after oxidation, hydration, and oxidation, and finally split into the 2 molecules of acetyl coenzyme A. And then the acetyl coenzyme A can be oxidized to the carbon dioxide and water via the citric acid cycle. Okay, and then the FADH2 and NADH can be can fit electrons into the respiratory chain. So this is a beta oxidation of fatty acid. We named this process as this process is a beta oxidation of fatty acid. Okay, so next let's see the energy yield from the fatty acid oxidation. In fact, oxidation of fatty acid can produce a larger quantity of ATP. Okay, so next we take palmitic acid. We take palmitic acid as an example to calculate the energy yield from the fatty acid oxidation. The palmitic acid contains 16 carbon atoms. Okay, and so after beta oxidation, the palmitic acid can be converted into the 8 molecules of acetyl coenzyme A. Okay, right? It can generate, so because it will have the including 7 cycles of beta oxidation and then result in produce 8 molecules of acetyl coenzyme A and 7 molecules of NADH and 7 molecules of FADH2, right, can be generated. And then the acetyl coenzyme A can be metabolized by the citric acid cycles, and the NADH, FADH2 can be oxidized by the electron transport chain. Okay, so after one citric acid cycle, it can generate 1 molecule of acetyl coenzyme A can generate 10 molecules of ATP, right? And 1 molecule of NADH can generate 2.5 molecule of ATP, and 1 molecule of FADH2 can generate 1.5 molecule of ATP, right? Okay, so if you count it all up, the oxidation of 1 molecule of palmitic acid can generate the 108 molecules of ATP. So, and then here you should notice that at the beginning, in the activation of fatty acid, you remember they consumed 2 molecules of ATP. So two ATP must be subtracted for the initial activation of the fatty acid. So the net gain of the ATP is 1. Yeah, I don't know if you remember here, the activation of the in the activation of fatty acid consumed 2 molecules of ATP, right? Because here you should notice that the ATP should provide two phosphate group. So that means that that means that we need 2 molecules of ATP. Okay, so that's the that's all for the fatty acid oxidation. Okay, and let's summarize. Okay, fatty acid oxidation in mitochondria is mainly happened. It occurs in the mitochondria and then result leading to the generation of larger quantity of ATP by a process called named beta oxidation. Okay, and the after beta oxidations, the acetyl after beta oxidation, this acyl coenzyme A can be cleaved into the acetyl coenzyme A. Okay, and the acetyl coenzyme A is oxidized in the citric acid cycle, and then generate the. This can be occurs in the starvation after exhaustion of glycogen stores. And the same situation can also be occurred in untreated type 1 diabetes. As we know, the patients with diabetes are inability to metabolize carbohydrate effectively, right? So usually they use they use fat metabolism. The fat metabolism is a main source of energy for these people with diabetes. So in this situation, the body has a high level of glycogen of a glycogen, right? You know the glycogen because because they cannot use the glucose effectively, right? So in the blood glucose level will be increased, and then the body has a high level of glycogen. So the in this so the adipose cells are releasing excessively amounts of fatty acid in response to high level of glycogen, and the activation of, as just now we have mentioned before, the glycogen can activate the activation of hormone sensitive lipids, right? So in this situation, the liver produces larger amounts of acetyl coenzyme A, which cannot be handled by the citric acid cycle. So next we will learn how to deal with this extra acetyl coenzyme A, and next we will learn the ketogenesis. Okay. So what is a ketone body? The ketone means the acetyl acetate and 3-hydroxybutyrate and acetones. Okay. So if we in our body we have generate extra acetyl coenzyme A, the hepatocyte can join to acetyl group, acetyl groups together to form the acetyl acetate, and which is partially reduced. Part of the acetyl acetate can be reduced to the hydroxybutyrate, and then they can also be converted into the acetone. And this acetyl acetate and hydroxybutyrate and acetone, they are known as ketone bodies, right? And ketone bodies, and this acetyl acetate and hydroxybutyrate can be released into the bloodstream because they are water soluble and can be transported in blood to extra hepatic tissues. So here you should remember the livers produce ketone bodies, but cannot use them itself as metabolic fuels. Okay, so that means the ketone body genesis, the ketone body can be produced in they are produced in liver, but they can but the liver cannot use them itself as a metabolic fuel. And here, this should use a okay. So let's see that just now I have told you that there exist three substances known as ketone bodies: acetyl acetate, hydroxy butyrate, and acetyl, right? Okay, so next let's see the ketone body formation. That means how to form these ketone bodies. Okay, and ketone bodies is forming ketone body formations occurs in the mitochondria in liver, and only in liver. Here you should remember. Okay, so first. Here, two molecules of acetyl coenzyme A join together to form the acetyl coenzyme A. And this reaction is catalyzed by the acetyl coenzyme A sialase. Okay, and then next, a third molecule of acetyl coenzyme A is used to form the hydroxyl coenzyme to form these 3-hydroxyl 3-methylglutaryl coenzyme A. That means HMG coenzyme A. And this reaction is catalyzed by the HMG coenzyme A synthesis. And then, after hydrolyzation, the HMG coenzyme A can be converted into the acetyl acetate, right? And result releasing another acetyl coenzyme A. And then this acetyl acetate is partly reduced to the beta hydroxyl butyrate. Okay, and then it can also this acetyl acetate can also be decarboxylate to acetyl, and this acetyl is a volatile solvent which is exhaled. For example, in untreated type 1 diabetes, in the patients with untreated type 1 diabetes usually have a high concentration of the ketone bodies in the blood, and these acetones give rise to the characteristic of fruity smells in the breath. Okay. And next, let's see the utilization of ketone body. Acetyl acetate can be used by the peripheral tissues. That means the ketone bodies can be activated to the acetyl coenzyme A, and this reaction is catalyzed by this succinyl coenzyme and acetyl acetate coenzyme transferase, and then the coenzyme A is then transformed from the succinyl coenzyme A to form the acetyl acetyl coenzyme A, and then the acetyl acetyl coenzyme A is splitted to form two molecules of acetyl coenzyme A by the sialase, and then the acetyl coenzyme A can be oxidized in the citric acid cycle. And here, beta hydroxyl butyrate. Beta hydroxyl butyrate is also utilized by being dehydrogenated first to acetyl acetate, and then can be converted into the acetyl coenzyme A. So here you should remember that the ketone bodies can only be synthesized in liver, right? But the liver cannot use the ketone bodies as an energy. And the ketone bodies in the liver, synthesized in the liver, should be released to the extrahepatic tissues. Okay. So in this ways, the liver does not generate and use up the liver can only generate, but it cannot use the ketone bodies at the same time. Okay. Okay, so next, let's see the physiological significance of the ketone bodies. The ketone bodies are just now we have learned as you see, the ketone bodies are very small, right, and they can diffuse through the blood-brain barrier. So, but the fatty acid cannot transform through the blood-brain barrier. So the brain cannot use the fatty acid as an energy. So, and the ketone bodies is very important for the brain. During the starvation, at this time, the glucose supply is limited, right? So the brain can utilize the ketone bodies as an energy, and the ketone bodies can relieve to demand for the glucose. And in prolonged starvation, up to 40% of energy in the brain are generated from the keto bodies. So the keto bodies is very important for the brain, okay, especially in the starvation conditions. That is because you should remember that is because the keto bodies is small and they can be easily diffused from the blood-brain barrier, okay. And using of the keto bodies is also important as it spares muscle protein, which would otherwise be degraded at much higher rates to provide glucose, with detrimental effect on the organisms. Okay, and if excessive production of ketone bodies, it can lead to the ketoacidosis. And ketoacidosis is a potential dangerous conditions. And note that the ketoacidosis does not happen in the fasting or starvation, because in this situation, a small amount of insulin is present and exerts some control over lipolysis. But the ketoacidosis can occur in the patient with untreated type 1 diabetes because of the high level of glycagon. Okay, so we have finished the okay. Let's summarize this part. This part just know this we mainly talk about the ketogenesis, right? And here you should know the keto bodies includes the acetoacetate and 3-hydroxybutyrate and acetone. And the keto bodies are formed in the hepatic mitochondria. That means in the mitochondria in liver cells. Okay, and because in the liver cells, there is a high rate of fatty acid oxidations. And the second one should remember that the keto bodies are important fumes in the actual hepatic tissues, especially for the brain. That is because the brain cannot use the fatty acid as the energy. And the third one you should remember the ketosis is mild in starvation but severe in diabetes, especially in untreated type 1 diabetes. Okay.